about what is biosensor technology. Biosensor technology is an analytical device that provides information about a biological process through a transducer. As you can see that based on the picture that I provide in the slide is the biosensor structure. And the information from this biosensor technology can be both semi-measurable and measurable. It is a biochemical receptor that used to collect information and transfer it straight to transducer. And finally, it will send the signal to be read and recorded. An ideal biosensor should be in both three, which is temperature and pH independent, recyclable, and lastly is specific. The usage of biosensor is a lot in different type of field in the world nowadays, and I only highlight two different field, which is in the food industry and in the microbiology field, as this is the most used biosensor. As in the food industry, biosensor is used to analyze nutrients, to detect natural toxins and anti-nutrients, for monitoring of food processing, and lastly for detection of genetically modified organisms. As for the microbiology field, it is used to detect a target substrate, converts the detected signal to a quantifiable response in a physiological, electrical, and biochemical manner. Now I'm going to talk about the catalytic biosensor. The principle of catalytic biosensor is first, biological recognition, which is highly specific towards the biological analyte. This transducer detect and transduce signal from biological target to electric signal, which is due to the reaction occur. And the third one is this electrical signal are amplified and can be read in detector after processing the value that are displayed in the monitor. Now let's move on to the application. Due to their high specificity, enzyme-based biosensor represent a fast and continuous monitoring of analytes. The high specificity of enzymes enhance the ability to detect lower analyte concentration limits. This enzyme function could cause the direct measurement of the analyte or for oxidation or reduction of a molecule, which can be monitored electrochemically. Now, the advantage of this catalytic biosensor is first, high specificity of enzyme-substrate interaction and the high turnover rates of biocatalysts. Second is the abilities to detect a broad range of combustible gases. And the third is it is a continuous and real-time analysis. And the last one is reduce the amount of harmful emissions released into the environment, which is good for us. Now, there is also some limitation of this catalytic biosensor. One of them is the rapid loss of enzyme activity due to the interactions with the electrode surface. Warm-up time is long before can be used. A significant effect of humidity on the sensor output, the limited selectivity for specific enzyme and substrate, and lastly is sensitive to the changes of temperature from the surrounding.
Next, the affinity biosensor. The term affinity biosensor in this refers to a device incorporating immobilized biological receptor molecule that can reversibly detect receptor-ligand interaction with a high differential selectivity and in a non-destructive fashion. Involved in the kinetic, structure, and solid or liquid interface phenomena associated with protein-ligand binding interactions. The potential application area for which affinity-based biosensor technique show promise include clinical diagnostic, food processing, military or anti-terrorism, and environmental monitoring. Next, there are a few advantages and limitations of affinity biosensor. They have the capability to provide for diagnosis and monitoring of cancerous, cardiovascular, infectious, and genetic diseases by the detection of biomarker using affinity interaction. Next, the limitation. They will exhibit saturation limit as well, and this is a consequence of the material used in designing the sensor. If a signal is saturated, that means that the biosensor is at max capacity and cannot detect any more substances.
Next is unparametric biosensor. For principle and their application, unparametric biosensor measure the current flow between electrons when a redox reaction takes place. Their principle is based on measuring current generated by enzymatic or bioaffinity reaction at the electrode surface under condition of constant working potential. Glucose biosensor is one of the most investigated of unparametric biosensor. And basically, it has two generations, which are the first generation will measure concentration of analyte or product of enzymatic reaction. Second generation will act as electron carrier, and the last generation will continue as direct electron transfer between enzyme and electrode. For the advantage, it is high sensitivity, selectivity, low cost, and easily to integrate into continuous analysis system. While their limitation is the interface produced between the enzyme and the electrical contact generating a slow biocatalysis, and it will result to the response of analyte substrate unparametric become low.
Next, I would like to discuss about voltammetric biosensors. Voltammetric biosensors are a type of biosensors that utilize voltammetric technique to detect and measure analytes of interest in biological samples. These biosensors are designed to combine the selectivity and the sensitivity of the biological recognition elements such as enzyme, nucleic acid, and also antibodies with the electrochemical principle of voltammetry. In the context of biosensor, the voltammetry provides a means to convert biochemical interaction between the recognition element and the analyte into an electrical signal. The general principle behind voltammetric biosensor is this biosensor involved in immobilizing the biological recognition element on an electrode surface. This can be achieved by various techniques such as adsorption, entrapment, covalent bonding, and self-assembled monolayers. Voltammetric biosensor has been used in food industry for years for food analysis processes. Voltammetric biosensor are utilized in food industry to monitor the quality, safety, and authenticity of the food products. They can detect contaminants such as pesticide, antibiotic, mycotoxin, and also any allergens. Voltammetric biosensor can be used for monitoring fermentation processes and determining the quality of the parameter of the beverage such as alcohol content and also acidity. Next, the advantages of voltammetric biosensor is it has high sensitivity, high selectivity, a rapid response, a wide dynamic range, and also minimal sample requirements. For limitation of voltammetric biosensor, it has limited analyte specificity, a limited shelf life, degradation and stability issues, and lastly, it has possibility of interference from complex matrices.
Impedimetric biosensor. Impedimetric biosensors measure the changes in charge conductance and capacitance at the sensor surface as the selective binding of the target occurs. The impedance of electrochemical cell surface is investigated in impedance spectroscopy, which is in proportion and dependent with the concentration of analyte and application of the sensor. It is an ideal tool for observing the dynamics of biomolecule interactions. Impedance technique as one kind of the electrochemical biosensors has been proved to be a promising method for foodborne pathogenic bacteria detection due to its portability, rapidity, sensitivity, and more importantly, it could be used for on-the-spot detection. Impedimetric biosensors have a simple structure and can be used to rapidly and sensitively detect and measure hazards in food. Impedimetric biosensors detect and measure target molecules by transducing biochemical reactions on their surface to electrical signal outputs responding to a sinusoidal electrical signal input. Next, the advantages of impedimetric biosensor. Firstly, high sensitivity, non-invasive measurement techniques, rapid and cost-effective technique, direct detection of biomolecular recognition events without using enzyme labels. Next, the limitations of impedimetric biosensors are limitations to detect small molecules, reusability, sufficient stability for repetitive measurements, short shelf life, and high probability of cross-sensitivity. In conclusion, biosensors are classified according to their biological receptors or transducer elements. Imperative utilization of biosensors has acquired paramount importance in the field of drug discovery, biomedicine, food safety standards, defense, security, and environmental monitoring. This has led to the invention of precise and powerful analytical tools using biological sensing element as a biosensor. These novel analytical tools are small devices that use a biological recognition system to investigate or detect molecules.